Alrighty, so this is the workbook video for B2.2 and B2.3, so cell compartmentalization and cell specialization, differentiation, development, cell structures. At the top of my head, I couldn't tell you exactly what it's called. I did do something different, so please don't freak out for my friends who I know really enjoyed a color with me. Per request, because I had a lot of people telling me, oh my gosh, Miss White, I really like the videos, but it just takes so gosh darn long, and half of it's coloring. I heard you, I feel you, like, listen, I gotcha. So what I did is I went through and I pre-colored some of the stuff, just to try to, what is happening? Sorry. Just to try to save us a little bit of time. Of course, as I go through it, I'm going to explain, like, what the color coding means to you still. That way you're not like, what the heck? But I figured it might help us, like, go a little faster, maybe? I don't know, we'll see. So your first page is all about organelles as discrete subunits of cells that are adapted to perform specific functions. If you know, we did the little compartmentalization sheet where you looked at specific organelles and how they were adapted. You looked at, I think it was mitochondria, your nucleus, free ribosomes, Golgi, chloroplast, and bound and unbound ribosomes. Did I already say that one? Mitochondria, is that the one I'm missing? That might be the one I'm missing. Anywhoosies, organelles are essentially just small structures inside of prokaryotic and eukaryotic cells. I made prokaryotic purple, P for prokaryotic, P for purple, and eukaryotic red because it was, I don't remember. Oh, yes, I do remember. Because then I could have a fun warm color gradient for things that are organelles, which, spoiler, are mainly in eukaryotic cells, and then a cool color for things that are not organelles, which are almost exclusively in prokaryotic cells, I'm pretty sure, unless I'm crazy. No, I guess cytoplasm, cytoskeleton, cell wall. Those are in both. But the goal of having these organelles is that they are adapted to perform one or several important functions for your cells. What I did on mine is I went through and I highlighted all the ones that are exclusively in eukaryotic cells in red, exclusively in prokaryotic cells in purple, which I think is just the nucleoid. And then what's not showing up well, because of my unfortunate color choice, is I double highlighted ones that are in both, so ribosome, cytoplasm, cell wall, cytoskeleton, flagella, just so that it's like a quick reference and then you can find them below. Organelles are organized into, well, I guess they're not all organelles, which is why I don't really like that this says arrange the following organelles, because they're not really all organelles. It should be arrange the following structures, but cellular structures are divided into first organelle, which is all the stuff that's going to be at the top, that's why it's marked in brown, and then not organelles, so structures that either aren't discrete enough or don't have a specialized enough function, or just kind of like, I don't know, like loose. Those structures are not organelles versus organelles. And then within organelles, there's like a hierarchy of, almost of complexity, arguably, of organelles without membranes, organelles with just a single membrane, and then organelles that have a double membrane for one reason or another. For some of them, it's because of that engulfing and endosymbiosis. For some of them, it's the invagination and endosymbiosis, but none, single, or double. All right. Don't think you escaped all the colors in the video, though. It's still colored pen time. Bum-ba-da-dum. I guess I'll label the top one first. We'll label this as organelles. It always cracks me up when the freshmen go organelles. Organelles. Because all of these are organelles. And then we'll fill in our no membrane organelles. I hope the yellow shows up on this gray. Fingers crossed. We'll see how it goes. So your organelles without membranes, does that show up? Oh, good. Your ribosomes, 2 subunits, but not one of them has a membrane. Your centrioles, Those are like the little pasta-looking ones that do the spindle fibers. Microtubules. And your nucleolus. Your nucleus has a double membrane, but your nucleolus itself, no membrane. It's just loosey-goose in the nucleus. Your things that have a single membrane, so one phospholipid bilayer, like looped around, are your vacuoles. Do you remember? Are mainly in plants. Your vesicles. Your rough ER, which is kind of a weird one because technically it's kind of an extension of your double nuclear envelope membrane, but because the rough ER is technically just like the cisternae, they individually each have a single membrane, even though they're kind of technically part of just a gigantic chaotic double membrane. They're kind of a wacky one. Your smooth ER, your Golgi. All right, I'm also going to warn you, in the packet, it calls your Golgi apparatus. I'm not writing apparatus every time. It's just going to be Golgi. You know what I mean. I'm called the Golgi, like a goofy eye. I crack myself up. Your lysosomes. And then flagella's kind of a weird one. When you see flagella in prokaryotic cells, it doesn't typically have a membrane. But when you see flagella in, like, the one eukaryotic cell that has it, which is a sperm, it is covered by a membrane, but it's a single membrane, so we're going to put her kind of weirdly in the middle, because she's kind of an exception. Some sources will also not consider your flagella an organelle. IB says it's an organelle, but says it's membrane-less, but then they also give you the sperm cell, which clearly has a membrane around its flagella, so I don't know. It's a wacky one. She's a little special. And then for double membrane, we have the bad boy himself, the nucleus. Get prepared to talk about this nucleus as double membrane. It's going to come up a lot. Your mitochondria, because of endosymbiosis. Your chloroplast, again, because of endosymbiosis. And your chromoplast, which is just like a pigment-producing cell. It's similar to like an amyloplast, which is like a starch-producing cell. Your chromoplast also has a double membrane because of endosymbiosis. Oh, you know what would be fun? If we mark them on here. Okay, hang on. Nucleus. Where's the mitochondria? Mitochondria. Oh, it looks like I scratched it out. I didn't mean to do that. There's no chloroplast. That's lame. Is there a chromoplast? No. Okay, maybe this is not as fun as I thought. There's not as many of them as I thought there would be. I'm kind of upset. All right, on to the next one. Vacuole. Oh, shoot. Golgi. Your ERs. Both of them. Where did they even label the ERs? Where's the label? Why is it over here? Lysosome. I guess your membrane's technically a membrane. Oh, I guess a peroxisome would have one. I think that's it. Oh, shoot. Nucleus, nuclear envelope. And then no membrane. This yellow is not going to show up on this picture. Oh well, there was an attempt. Your nucleolus, your microtubules, your, oh, where is it? Does it have centriole? They don't even have centriole? I'm so sad. And then your ribosomes. I'm not doing all of them. There we go, that's enough. And then, the ribosomes here too. Close enough. Oh, and flagella. It's a prokaryotic cell, so poor flagella is gonna have none. There we go. That was fun for me. Okay. Things that are just not organelles, like period, they don't apply. For whatever reason, they're not an organelle. Your cell walls. Your cytoplasm. I didn't think this through. How am I going to color in the cytoplasm blue? Oh, well, it's too late. Your cytoskeleton. And your nucleoid. This is just like loose DNA. Here's the nucleoid. I'm not going to color the cytoplasm, but like we'll just circle it here. We know. Your nucleoid, cell wall. Not the capsule, the cell wall. It's the capsule technically too, but whatever, it's fine. And then I don't think we see cytoskeleton. We see some microfilament, I guess. Yeah, I guess microfilament. That looks really stupid. Okay, you know what? There was an attempt. I'm happy enough. Alrighty. And so when we're looking at organelles as discrete subunits, your eukaryotic cells obviously have more organelles than prokaryotes, and therefore are said to be compartmentalized. I always think of like a dinner plate with compartments, or like when you're packing a suitcase. Compartmentalization just means that they have specialized areas for structures within the cell body which carry out certain functions. It's usually your organelles that does it. Prokaryotic cells don't have organelles, so they really don't have much compartmentalization. And although you might be thinking, oh, that sucks, poor prokaryotic cells, the benefit that prokaryotic cells get is that they can carry out cellular functions a lot faster. And typically more energy efficiently. At the sacrifice of being less complex processes. I don't know why there's a monkey here. All right, on to the next one. See, look at me go. It's already pre-labeled partially. I was really excited about this. Can you tell? Alrighty. So this is like your advantage of separating your nucleus and your cytoplasm into separate compartments, which of course only happens in eukaryotes because they're kept inside your membrane-bound nucleus. It allows for editing of your DNA before, or I guess editing of your mRNA before it goes through transcription, or I guess after it goes through transcription, before it goes through translation on a ribosome, which allows for removal of introns, editing of genes, more complex genes, more gene options, and typically like the ability for them to be modified more after translation, and they can make like a wider variety. Whereas with your prokaryotes, they have no membrane around their DNA, they just have that nucleoid. So your proteins are synthesized and then immediately your mRNA is transcribed into proteins. And so, itch. I gotta itch my finger, I'm so sorry. And so, in your nucleus, woof, you have your nuclear envelope, which is this beautiful, this is not gonna show up well, this beautiful little outside area here. It separates your DNA, which is just loosey-goose in here, from everything else. In your prokaryote, like your DNA is just loose in here. Like this is mRNA. I don't know where the DNA is not pictured, but this is your mRNA. Immediately being attacked by ribosomes. They can't run. Whereas mRNA can be separate from ribosomes. Yes. And so what this separation allows, first of all, it allows for protection of your DNA. It's less likely to get damaged if it's in like its own little special bubble. And second, and most importantly, it allows for editing and modifying. Oh, I don't know what happened there. Modifying of mRNA before translation it allows for more diverse proteins and reduction of mutations. And so then down here, you have little cell models of the process of transcription and translation. I went ahead and I highlighted just the structures and steps that are only in eukaryotic cells. You'll notice some of the things they have in common. They both have DNA, although in this case it's inside the nucleus. And so it starts with your DNA, becomes mRNA, mRNA, goes to a ribosome, and boop, makes your protein. But in this case, your mRNA edit, it gets edited before it comes out. So you have your precursor mRNA, which is like the draft version. It's your draft. And then this is your final, which is what eventually leaves. Whereas in your prokaryotic cell, you have your DNA goes straight to mRNA slash ribosome slash protein. There's no delay. It's immediate. As soon as that bad boy's made, it gets produced, which again is much faster and more efficient, but also it limits you in what proteins can be made. But don't think that's all. That's not the only advantage of your nucleus. Oh, before we get to the nucleus, because you all did so much like research filling in that page on compartmentalization and specific examples, fill this one in using your note sheet. I'm gonna skip it for the video because you all like already did this. I don't feel like we need to spend time on this again. Okay? Oh, they really take a while before we get back to the nucleus. I fully forgot that we have to go through the mitochondria first. All right, mitochondria first then. And I'm gonna warn you, your mitochondria, I know it's not gonna make a ton of sense. I'm sorry. I know that we haven't learned about cellular respiration, so a lot of the steps in cellular respiration aren't things that you're super familiar with. It's okay. Bear with it. As long as you know in general what each part does and like why it's important, then as we learn about cellular respiration, you'll be like, oh, that's what Miss White was on about. You're right. That does make sense. I promise. And so your functions of your mitochondria are ATP production, through aerobic respiration, And fat hydrolysis, or like breakdown of fat. Only if it's used as energy in the cell. And so just to help make this make a little bit more sense, cell respiration, or like aerobic respiration, in 3 steps. Your first step is glycolysis, which happens in the cytoplasm. So not even in the mitochondria. It's also just what anaerobic respiration is, or respiration without oxygen. Your second step is your Krebs cycle. And then your third step is the electron transport chain. Abbreviated as ETC. And so those are the functions. That's what happens here. That's why we need this. Now let's look at the structures that it has to help it do that. So, oh, see, this is one that I should have highlighted ahead of time. I don't remember why I didn't, genuinely. So, you first have, I guess we'll use this one, your outer membrane. Boop-a-da-doop. Which is just like the outer layer of your mitochondria. Ugh. This image is also kind of wacky because it has half like a diagram and then half an electron microscope scan. So there's your outer membrane. I guess here it would be like just this outside part. Outer membrane. Careful, 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 careful. Okay, that's stressful. And so your outer membrane is, oh, I shouldn't write it in the same color. That's going to get confusing. Oh, so the other pink. It's ever so slightly different. It's just the outer boundary. Of your mitochondria. It helps provide evidence for endosymbiosis. After your outer membrane, you have your inner membrane. Which is exactly where you'd think it would be. Slightly inside the outer membrane. Oh, why did I pick this purple? Oh no, okay, you can see the difference. Careful, Jessica, careful, Jessica, careful, 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 careful. Ooh. Okay. Your inner membrane's got like a laundry list of things to say about it. So I'm going to write it away from the word inner membrane. But it's color-coded, so you'll know what I mean. So your inner membrane, I'll put a little star next to it. It is your inner phospholipid bilayer. It contains membrane proteins. Which, like, are electron carriers. It helps to accept electrons. And pump positive hydrogen ions from matrix into intermembrane space. It's like the space between the inner and outer membrane. Ooh, I don't know how I'm gonna put that bad boy on here. All right, just gonna have to get ugly. Just gonna have to do brown for the intermembrane space. What is this? What that even? Okay. Oh, you can't even see the brown. Okay, maybe it'll have to be black. That's so boring. Oh well. Well, this is the space between the membranes. It's your intermembrane space. I guess it... Oh, it's so ugly. Okay, it's fine. So your intermembrane space... This pen's squeaky. It's making me so upset. It is quite literally the space in between them. Space between inner and outer membrane. The word membrane getting cut off? No. Excellent. Accumulates positive hydrogen ions to create high concentration gradient for chemiosmosis. Again, I know that there's going to be parts in here where you're like, what the heck? Just know that they all serve a function for cellular respiration. All right, next part. Oh, what's my best plan of attack here? We'll do our cristae, or one singular crystal. These are these little infoldings of your inner membrane. They're like what makes it look stripy on a little scan. They're kind of technically part of the inner membrane, but whatever, it's fine. All right. Where will we put this? I guess we'll put it with inner membrane. That way they can be together. So your cristae. Oh, why did I write that one but not write inner membrane? Oh well, it's too late. They are infoldings of the inner membrane. They increase surface area for the electron transport chain, because that's like membrane bound, and so it needs extra space in the membrane to store those. What do we have left? Oh good, we only have 2. So next we have our matrix. Let's make the matrix green. Boop. Doo-doo-doo. Just like the inside space. elegant. Oh, almost done, almost done. There we go. Oh, where's the green? Not the light green. We'll do the dark green. So it is just the fluid filled space where Krebs cycle happens. Notice that having your second step and your third step right next to each other, that's a big adaptation. So allows close proximity between ETC and Krebs. It also has enzymes that are specific to it, which help gradually boop, 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 scooch, oxidize acetyl-CoA to produce electron carriers like NADH. Again, it'll make more sense. I promise. Alrighty. What's left? Oh, just ribosomes. Easy money. Okay, what color are ribosomes going to be? We'll make them dark blue because I think that's the only thing that'll show up. Oh, well, okay, we'll just circle it. All right, there's some ribosomes. You get the gist. So your ribosomes, they produce proteins needed in mitochondria. Mainly enzymes, coenzymes, membrane proteins, and proteins for the electron transport chain. By having its own way, I'll scooch it. By having its own way to produce proteins, it's a lot more efficient. It's another thing that helps with proximity between like where it needs to be used and where it's being made. Alrighty. And that's it for mitochondria. On to the next step, which is, again, one that I'm sure you're like, what the heck? But it'll be good. So your chloroplast, shocker, is where photosynthesis happens. Just to help, photosynthesis in 2 steps, just to help it make a little bit more sense. Where am I going to put this? I need space to write, so we'll put it up here. So photosynthesis in 2 steps. First step, light-dependent reactions. Second step, your Calvin cycle. Oh, geez Louise. Or your light-independent reactions. And then, of course, you have all of these things. All right, what's the best plan of attack here? think, Jessica? I don't really want to make it blue, but I think it might have to be blue, which is unfortunate. I mean, all of them are just kind of in the thylakoid, which is going to make labeling it weird. It's okay. You know what? It's fine. We'll make them green, but it's just going to be weird, and I'm sorry. So your thylakoid, which is just these, like, little discs. So, like, these little discs. And they're little connectors. Jeez Louise, trying to color all these is going to be a mess. I'm not going to color the tops of the ones in the electron scan, just because otherwise, like, how are we supposed to color the stroma? It's just not going to work. Alright. And here's the thing. Your granum is also kind of your thylakoid. Your granum is just like a stack of thylakoids. So, I'm just going to draw it like that. Here's one granum. Just a stack. And then we have all kinds of things about our thylakoids. Let me get the light green. So your thylakoids have two different things that help it out. It has your thylakoid space, which is like the inside of a thylakoid. And it's a small, restricted space. It allows for rapid accumulation of positive hydrogen ions needed for production of ATP by flowing through ATP synthesis, or synthase, I'm sorry. How do synthase, which is an enzyme. Okay. Then you have your thylakoid membrane. Scoochy, scooch, 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 which I feel like they should be on the same side. Yeah, okay. So then you have thylakoid membrane. Here's membrane, which is just the membrane around a thylakoid. And so it has a large surface area, which I'm going to shorten to SA, because there's many thylakoids, oh geez, stacked on each other. Oh. we're going to do a little parentheses granum. That's what the stacks are called. And this is where your photosystems are located, and where light is absorbed. And light is needed for light dependent reactions. They depend on light, duh. Scooch. Maybe I should have scooched this to the other side. This is like a lot. Sorry. It also contains the electron transport chain. And having a lot of membrane means a lot of ATP. And NADH are produced during light dependent reactions. And so again, that's like a proximity thing. Where it absorbs light is also where it makes, like where it makes and what, like where that reaction takes place. Where it makes what that reaction needs and where that reaction takes place. All right. And then next up we have stroma. Ooh, my foot itches. I don't want to make it blue. That's going to blend in too well. I guess stroma can be the ugly pink. I'm sorry, stroma. I'm sorry, stroma. It's just like the space between the thylakoids, which, listen, I know in a weirdly 3D model like this is questionable to color. So your stroma, woof. It's where your Calvin cycle takes place, where your light independent reaction takes place. Again, close proximity, having what's produced right next to what, like where the reaction occurs. It's where NADH and ATP are used to produce sugar. And it is just the fluid-filled space around them. It's fluid-filled and contains enzymes like rubisco, needed for the reaction. So again, able to have the enzymes and the reactants in close proximity and high concentrations, able to have what's needed right next to where it needs to be used. Woof. You also have your inner and outer membrane. Specifically, your outer bilayer membrane is important. Woo! I tell you, organelles are like not my most favorite thing. Maybe I should have done the outside green. Oh well, it's too late now. And your outer membrane, similar to your mitochondria, is just outer boundary and evidence for endosymbiotic theory. And that's it. Thank goodness. Getting a little sick of these. Alrighty. Oh, finally. Now we get to talk about the nucleus again. I missed her. Alrighty. So your nucleus, it is a double membrane, and this double membrane is really important. Your nuclear membrane requires transport of large molecules in and out, and your double membrane essentially allows for bigger pores. It also allows for the nuclear membrane to be broken down into vesicles during like cellular division. What I didn't label on here is your nuclear pores. So your nuclear envelope, it's just the outside double membrane, your nuclear pore. Then you have nuclear pores all around. The way that they drew the chromatin with the nucleoid, it makes it look like, nucleolus, it makes it look like it's hairy. But you have nuclear pores. On the outside, they look like little silly flower speckles. And if you notice, it is connected to your ER, which is pretty cool. And so what needs to be transported in and out of your nucleus? Hang on, let me scooch things around on my desk real quick. Move. Okay. So you need to have enzymes for replication, / transcription, mRNA, hormones, ATP, transcription factors, and of course your rRNA made in your nucleolus. And so how does the double membrane of your nucleus help with that better than a single membrane? So it, first of all, allows for larger pores. Larger pores than a single membrane could support. It allows for your nuclear envelope to be directly connected to your ER. Which just makes it more efficient. And it allows for nuclear envelope to break down for mitosis / meiosis or cell division by breaking apart into vesicles. And the reason for that is so your chromosomes can separate. Oh my god. It's my phone ringing. What the heck? It's my brother. We're gonna hang up on him. Get out of here. He doesn't need to hear from me. He's probably gonna call me back. He's gonna be like, what the heck? Why is Jessica not answering? Why is she not home? Dramatic. Alrighty. And that's it for the nucleus and the nuclear envelope. Now we get to talk about membrane-bound ribosomes that are bound to the ER versus free ribosomes. And so ribosomes are composed of RNA and proteins. The complex is 2 subunits, a small one and a large one. Their main function is just to produce proteins through translation. And your 2 ribosomes, the main difference isn't in their structure, what they're supposed to do, like how they do it. It's in where the product is intended to go. So for membrane-bound ribosomes or ribosomes attached to the ER, they attach to the outer membrane. That's why when you draw or see your ER, you see it with like little speckles. Those are your ribosomes. And its main goal is to make proteins or polypeptides that go through the endomembrane system, usually for export through, from the cell. And so they either can stay in the ER, go to your Golgi and be modified, and then from your Golgi go to a secretory vesicle and leave the cell, go to a lysosome, digest something, and then leave the cell, or go to your plasma membrane to be like a membrane protein. Your free ribosomes, they're just ribosomes that don't ever bind to your ER's membrane. They just stay loosey-goose in your cytosol. And so they finish the protein in the cytosol. It either stays in your cytosol or cytoplasm and goes and does whatever there, or it goes into an organelle, so it either goes back to your nucleus, your mitochondria, your chloroplast, your peroxisome. Although your mitochondria does have its own ribosomes, there are some things that it does need more of that can come in through the cytoplasm. Okay. So it's less about the structure of them and more just about the pathway they take. I really kind of hate the way they did these pictures because to me personally, I feel like they should be swappied, but whatever. And so you have your free ribosomes, which are just loose, they're not bound. And then if you look, this is your ER here, and attached to the edge are your bound ribosomes. And then here you have your ER, and then on it, these little speckles, these are your ribosomes. Oh, I have the green one out, but I guess I need the orange one first, huh? Tisk, tisk, Jessica. So your free ribosomes, are they surrounded by a membrane? No. And that's not to say that they have a membrane. That's to say that they are like within something with a membrane or bound to a membrane. Composition is our RNA and proteins made in your nucleolus. They have 2 subunits. Your small one and your large one. So when they do protein synthesis, the like RNA kind of goes like through the middle, almost like that looks so dumb, like receipt paper. We'll call that mRNA. Location in the cell, cytoplasm, and potentially the RER. And then their function, produce proteins to function in cytoplasm or organelles. They stay inside, stay in the cell. It's mainly enzymes. And then for your rough endoplasmic reticulum, is it surrounded by a membrane? Yes, it is a membrane. For composition, it is flattened sacs called cisternae. It's the same thing in the Golgi of membrane, a network of tubules, and channels for transport. Tubules / channels that extend all over cell. It has ribosomes attached to membrane. Location in the cell, it's closer to the nucleus. It's in the nucleus region. Oh, where do you live? Closer to Gaithersburg. Like, closer to the nucleus. I'm sorry, I'm cracking myself up. And so it produces proteins for endomembrane system, parentheses, sent to Golgi for modification. Ultimately, they usually become lysosomes or get secreted. They're like outside. Leave the cell. Get out of here. Be gone. Speaking of the Golgi, there it is. Get excited. And so your Golgi apparatus, again, it's made up of those same flattened sacs of cisternae that are stacked up on top of each other. They have two sides, your cis side, which is your receiving side, and then your trans side, which is your discharge side. You can tell the difference because your cis side will always face the ER. Your trans side will usually face like the membrane. And it's basically just like a big loopy membrane that modifies and packages like polypeptides. And so in our little diagram here, if here is the ER, your proteins would go like woo and make their way through and then leave. And then here you'd have your like cell membrane. Because this is your trans phase and this is your cis phase. So these would be not outgoing transport vesicles, outgoing secretory vesicles. These would be incoming transport vesicles. I'm satisfied with that. But so it processes and collects and packages and distributes polypeptides from your RER, enzymes in cisternae, modify polypeptides, like example, adding carbohydrates, to like glycoproteins, some of the products are transported in vesicles to the plasma membrane and secreted. Examples like digestive enzymes, in the pancreas. Yay! Oh shoot, there we go. All right. Oh, wrong way. Bum-ba-da-dum. And so here is your endomembrane system, which I hopefully at this point you all are again pretty familiar with. Get this out of here. And so your endomembrane system, bum-ba-da-dum, has a series of steps. And so we'll do a little magic moment here. I color-coded it at the top, starts at your RER, protein is made, goes to a transport vesicle to the cis side of your Golgi, makes its way through the cisternae, gets modified, leaves through the transphase in a secretory vesicle to the cell membrane where it gets exocytosised. And so your polypeptides made in, oh, that was already open, whoops, rough endoplasmic reticulum are transported to the cis side of the Golgi via a, what color is this, orange? Transport vesicle. That's step one. Then processing in Golgi is sequential as proteins move through your cisternae towards the trans side they are modified and changed. Once processing is complete, your Golgi organizes proteins and a, oh, what color is secretory vesicle? Pink? Red? What is that? Red? I think that's supposed to be red, but it really looks pink, so we're gonna make it pink. And a secretory vesicle buds off the Trans-side. Secretory vesicle formed, takes the product to the cell membrane to be secreted via exocytosis woof. Close all these gosh darn pens. Oh no, I hear them coming to take my trash. They're gonna be like, what the heck is this lady doing? All right, how many pages do we have left? Stem cells. Hang on, I'm doing math real quick. Stem cells, stem cells, stem cells. And then this part we started in class. Okay. Alrighty. So I think the vesicles will be the last thing we cover today, mainly because we talked a lot about stem cells, and I think that you all can probably figure that part out. So we'll just talk about Clathrine. Hello.
Yeah.
And so your vesicles, they're fluid-filled sacs composed of a single phospholipid bilayer, which surrounds the fluids and solutes inside. They move materials through endocytosis out of a cell. Oh, I'm sorry. They move materials into a cell through endocytosis, EN, enter, and out of a cell through exocytosis, EX, exit, but can also do internal transport of substances. And so oftentimes they do transport from the ER to the Golgi and finally to the membrane, like seen above. Because they're made of a phospholipid bilayer, or I guess a single phospholipid bilayer, they have your hydrophobic regions and your hydrophilic regions.
Oh, that's good.
If you remember from Origin of Life, spontaneous formation of vesicles is how they think that a lot of life started. And so, what was I going to say to you? I'm so sorry. In vesicle formation, there is protein that helps it form. And so, sorry, I got really distracted. So this is your vesicle. This is your final vesicle. And the protein that helps it form is Clathrine. And so this is like a molecular model of Clathrine. This is like a little 2D model of Clathrine. And so how does this protein help the vesicle form? So it supports fusion of membranes when I really give this up. Vesicle is made. Clathrine molecules bind to each other, forming a coat. It helps plasma membrane become indented and detach to vesicle. So it essentially, like, this only happens in receptor-mediated vesicle formation. So you have your receptors, you have your cargo bind, you have your Clathrine here, your clathrin coat forms around it and then starts to kind of pinch in. And then once it kind of squeezes it off and pinches it off and it like leaves, it helps to seal this membrane in. And then once it's not needed anymore, once your vesicle's formed, it just straight up dips. And that's pretty much it for clathrin. Let's highlight the clathrin. So, whoa, careful. Clathrin. Oof. Clathrin coat. Clathrin coat. Clathrin, 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 clathrin. I'm gonna have to tell my friend Catherine that there's something called clathrin. Alrighty. I don't know, should we do stem cells? Should we? Alright, if you watch this, just know, I'm not going to do stem cells today. We'll do stem cell, I'll film the video for stem cells. What am I doing Monday? Anything? Monday I have a department meeting. Ew. Well, I'll try to film it Monday, and if not, Tuesday. And so the packet won't be due until Wednesday of next week on SAT day. So, spoiler, if you watch this all the way through. If you don't, and you're like, oh my gosh, I have to finish the whole packet by Monday? Crazy. I don't know what to tell you. Sorry.